Pat also hear me? Good. Excellent. So I'm actually going to end up probably where Anna ended up as well. So in recommendations for research project and what type of markers to use, but I'm going to fly in that direction, well, from a different direction. So I'm not starting with with lab work, but I'm going to give a very brief overview of phylogenetics, so phylogenetic methods and the kind of data you use for that. I mean, there's much more to be said, but I won't keep you busy for too long. So I've called it recent development in molecular phylogenetics. So I'm really going to give kind of conceptual overview of phylogenetics. Just briefly to introduce myself, this is where my academic career has taken me over the past 30 years. I did my PhD at Utrecht University in the Netherlands, then spent a year and a half, actually where Anna is now, in Kew Gardens in London, then went back to the Netherlands to Wageningen, and since 4.5 years I've been in Belgium at Ghent University, where I'm leading the lab of systematic and evolutionary botany. So my research specialty, my pet group, so to say, has been Ananaceae, a plantopical family of trees and lianas of around 2,500 species, of course, worldwide in essentially tropical rainforests. So here are just a few examples from Africa and the Neotropics. But recently, let's say, round about the time I got to Ghent, roughly 4 years, the Convolvulaceae had entered my life. So it's just a brief hint to Convolvulaceae. And I'm mainly going to use examples from Convolvulaceae in my talk, although in one case I'll also refer, no, 2 cases, refer to Ananaceae. Anyway, so Anna is from Kew. I worked for a year and a half at Kew, and this bloke also worked at Kew, and actually he was quite important. He's a former director. He was the second director of Kew Gardens, Joseph Hooker. So he was director of Kew Gardens around about halfway the 19th century. And he had a good friend. Just as Hooker, his friend was also white-haired and had a beard, and of course he was Charles Darwin. And they were really best friends, not just any friend, they were really very close friends. And so Charles Darwin, he visited Kew Gardens regularly, and if he wouldn't come to Kew, because he liked to really spend most of his time in down the house at Ghent, his home, they wrote letters. And so this is a letter from July 1879 that Charles Darwin wrote to Joseph Dalton Hooker, where he says that the evolution of angiosperms, the evolution of flowering plants, according to Darwin, is an abominable mystery. So that is what Darwin thinks of the evolution of flowering plants. Not just the evolution, but especially the fact that flowering plants for a long time, or for a very long time of course, are absent in fossil records. And once they appear in the fossil records, bang, they're all of a sudden all over the place. It's not just that they gradually started appearing, no, but once they're there, they're massively there. And that fact, a massive, all of a sudden appearance of flowering plants, that was the abominable mystery to Charles Darwin. And of course, we now usually tend to describe this phenomenon of an all of a sudden burst of lineages as a radiation, right? So, and even not just the angiosperms as a whole, as a complete clade of flowering plants, but also within the angiosperms, there are numerous examples of genera or family, which sort of, the evolution of which follows more or less the same pattern. All of a sudden appears a rapid radiation of lineages. And this is also true for Ipomoea. So on the right you see 2 pictures of specimens of Ipomoea that we collected last year on a collecting trip in Kenya. And in 2019, a group of researchers, mainly from Oxford University, in the UK published a large analysis of phylogenetic analysis of Ipomoea. And so Ipomoea is the genus that also includes Ipomoea batatas, the wild from which the cultivated sweet potato has been derived. And the authors say about the evolution of that genus Ipomoea that and you see since the origin of Ipomoea significant increases in net diversification rates comparable to the most iconic evolutionary, and that's the word, radiations in the plant kingdom. So even Ipomoea is a radiation. So, and radiations, and just like almost anything in plant evolutionary biology, can be studied, can be understood with phylogenetic methods. So that's what I'm going to talk about today. So how to do that, how to unravel these patterns. Phylogenetics, of course, I mean phylogenetic trees, I should say, they were around even way before we sort of inferred them using molecular data. So this is an example from a paper from Rolf Dahlgren, a very, very good Swedish botanist, and he made this picture in 1980 in one of his papers, and then all of these numbers refer to major, what we now would say clades, major groups of angiosperms. But this was very intuitive. So this picture, I mean, of course he was an eminent botanist, so he'd studied all sorts of plants and saw similarities and dissimilarities in morphological characters. And he used his knowledge of similarities and dissimilarities to come up with this picture. But still, you could say this is a rather intuitive phylogeny. There's no, let's say, hard method underlying it. It's just his knowledge. And that's a major shift that we've seen, let's say, mainly from the early 90s onwards. And since then, phylogenetics has become a formal discipline, with hard methods, with analyses, with statistics, and all of that. And I'm just now here putting up three kind of questions that you could ask about the abominable mystery of Charles Darwin. It's a possible hypotheses that have been driving the all of a sudden appearance of angiosperms in the fossil record, or on the evolutionary scene, if you wish. So one of the hypotheses is simply the origin of flowers itself. And so this is a very nice paper by Hervé Sauquet, a French botanist, but now in Sydney, Australia. He published with many co-authors in 2017 in Nature Communication. And the question is, what did the ancestral flower, angiosperm flower, look like? And you can so I'm not going to talk about what it looked like, but I'm just pointing out the fact that you can reconstruct this. So having a phylogenetic tree based on molecular data and having, in this case, lots of morphological characters of, how many petals, how many sepals, are they in whorls, or are they spirally arranged, etc., etc. And doing these analyses, they came up, and there's a kind of, let's say, impression or what it could have looked like down there in the bottom right, that could have been, the ancestral angiosperm flower. So the origin of flowers is very important for the abominable mystery as an answer. Another important answer that we know now has been imported in the evolution of flowering plants is the phenomenon of whole genome duplications, so polyploids. Plants tend to double their genomes much more frequently than animals do. And it's one of the hypotheses that this is why angiosperms are so successful, because if you double your genome, you've got double the amount of, genetic material, And of course, in the beginning, every function is being served in two ways, by one genome in two ways. But then gradually, you get the process that's called subfunctionalization. So the redundant bits of the genome are being recruited, to serve other functions, and you can do new things, so to say, with that duplicated genome. And so, so this is an example from a paper in Brassicaceae. So also in Brassicaceae, the mustard and cabbage family has been demonstrated that actually whole genome duplications are very, very well correlated to, let's say, important evolutionary advances in this family, and also for angiosperms in general. And then the third example is to refer again to the abominable mystery of Charles Darwin. I mean, first of all, you have actually to establish that it is an abominable mystery, and you can also do that with phylogeny. So this is a paper by Susana Mariel from Mexico, a number of years ago already, 8 years, where she did a very, very large scale phylogenetic and dating analysis. So the axis that you see there is not visible? No. Here. Yeah. The axis that you see there is a time axis. And so you see here are the gymnosperms, the outgroups, and then here this fossil time, nothing's really happening. Here is the common ancestor of the angiosperms. And you see that actually all the major lineages of angiosperms sort of, boom, as Darwin already knew from the fossil record, emerge in a relatively short span of time. Okay. Now I'm going to, so this was nice, what can we do, what can we learn from phylogenies, but I'm going to say a bit more like, well actually, how have we been making these phylogenies, how have we been inferring them? This paper is very, very, very important for that. This is in fact the first paper publishing the analysis of phylogenetic analysis of all angiosperms. Mark Chase is the first author. He's also a Kew Garden, so he just retired, but he's been there for a long time. And in '93, he had this absolutely brilliant idea. He was contacted by a molecular biologist, and this molecular biologist was interested in the function of the protein rubisco. And rubisco is the protein that captures CO2 from the air, so it's the start of photosynthesis. And so what this molecular biologist did, so I forgot his name, I don't remember who this was, but anyway. So this molecular biologist sent around primers for PCR, for Sanger sequencing that Anna has just been talking about. So he wanted botanists to sequence RBCL for all of their favorite groups. So this molecular biologist just would receive lots and lots of data, and he got those data. But then Mark thought, well, Mark is not a molecular biologist, he's like us, he's a systematic botanist. And so Mark thought, if you've got this data anyway, okay, for another purpose, but we might do other things with that as well. We might do phylogenetic analyses. So this paper from 1993, it's exactly 30 years ago, as I said, it's the first phylogenetic analysis of 500 species in this case of flowering plants. So this is the very, very first hypothesis for phylogenetic relationship based on non-intuitive methods, on repeatable methods. Right, then a few years later, 1998, I mean, I was there in those days as well. I started my PhD in '93, in '98 I finished, and in '98 I actually moved to Mark Chase's lab at Kew Garden. So, and there was a buzzing era, really, with lots of new discoveries about phylogenetic relationships in angiosperms. And this paper was published in '98, and that was published in Nature. And what was new in this paper is that for the very first time, so this is 25 years ago, a phylogenetic analysis was published for flowering plants with representative genes of all 3 plant genomes. I mean, Anna just talked about nuclear genes and chloroplast genes, plastic genes, but I'm sure you all know that also the mitochondria, plant mitochondria also have a genome, right? So plants have 3 genomes, and so this analysis had 5 genes, but all 3 genomes being represented. And what was new in this paper? What was new in this paper was that for the first time, with good statistical support, Amborella was pinned down as the sister group to all other angiosperms. Mark Chase in those days wrote, well, he still does, but wrote lots of papers on, how also, not just on what are the phylogenetic relationships like, but also how to do this. And I've always liked this paper very much. So what's shown here is phylogenetic trees based on individual markers, so RBCL, which is a chloroplast gene, ATPB, that's another chloroplast protein coding gene, and 18S, that's part of the nuclear ribosomal DNA, right? And so, and these then, here are some bigger groups of angiosperms, bigger clades of angiosperms. Now, the take-home message of this figure is that if you run analyses of individual markers, then you barely, you may get hardly any support, statistical support, bootstrap support for your analysis. However, if you, in this case, combine those three, so 18S, ATPB, and RBCL, if you combine those in a single analysis, then you all of a sudden get some serious support, which is indicated here by these blue, by these black dots. And so these dots indicate well-supported clades. So the message in this paper was just combine data. That's good. And so they write, it should be expected that different categories of DNA sequences should be subject to varying patterns of non-historical variation, whereas all of them should carry the same historical pattern. In other words, if you combine multiple loci, they all, to some extent, carry historical patterns, so the true phylogenetic pattern, but they also carry some, and I won't call it non-historical variation, they carry some noise. And the idea is if you combine markers, you reinforce the phylogenetic pattern, and you just kind of dilute the noise. And that's what you see in that figure, because you increase support. Now, this strategy is called data concatenation. So you glue your data partitions one after the other. And it looks like this. So this is from one of our own data sets in Indonesia. So here we've got, this is the program that Skeet, some of you may know that. So here are our species, and then you can see the divide there already. So in this case it's two loci, two genes or whatever. So this is the first gene, and here you've got the boundary. And then, so for example, for this species, Chrysosperma cauliflorum, this is the sequence of, say this is gene 1, and then nicely glue the sequence of gene 2 just next to it. That's data concatenation. And this has been the favorite this has been the favorite way of working for a very long time. But there is one very, very important condition. You can only do that. Okay, and I always like to ask my audience, who of you have been doing phylogenetic analyses actually? So do you know what is the first and foremost condition that should be met for you to be allowed to be able to do this, to concatenate your data? Yeah?
So can you repeat the question?
Oh, sorry. Okay, well, what is the condition? So there must be a condition, the condition must be met for this to be a reasonable approach. So to have sequences for gene 1 and for gene 2 and then analyze them together in this concatenated way.
So first we need to align the sequences.
But that's very good. But let's assume, well, that's the case. In this case, they are aligned already.
Yes. So I didn't get your question.
I'll answer it. Don't worry. The thing is, there should be no conflict in the phylogenetic pattern.
So you mean to ask, what is the need of concatenation in this case?
No.
The need is, well, the need I explained here. The need is more data. Is more.
What was your question? I'm trying to get your question.
Sorry. Okay. I'll answer it. So the thing is, you can only concatenate your data. So glue the loci one after the other. It's if each of these loci do not contain conflicting phylogenetic patterns. In other words, you can only do this if locus 1 does not significantly disagree with the phylogenetic pattern in locus 2. And if and you can have, of course, concatenate as many loci as you want. But this idea of incongruence, that is, I would say, that's lethal for your phylogenetic analysis. And I'll give some examples why actually concatenation is a very bad idea. Usually, I'm not saying always.
But if we need enough amount of variabilities also, not just similar patterns will give a robust result. That's why we do concatenation, so that we can get more amount of or enough amount of variabilities also.
Fair enough. But that's good. So that's what you say is good. You want more data, so you concatenate your data. But the thing is, my point is, you can only do that if you meet that criterion of non-conflict. And I'll show you where you get into trouble, right? And I'm building up my story here. So here is a short warning first, because if you could argue, well, I'm only, I don't know if that's the case with your project. You could say, well, I'm only aligning chloroplast genes, and the chloroplast is always inherited as one circular molecule. So conflict between individual genes on my chloroplast, that's not going to be an issue, because the whole chloroplast, the whole thing, is always inherited as one unit. That's what we thought too a number of years ago, but then we found this. It's a small cautionary tale, I should say. So we actually found, was a PhD student of mine, Mike Peary, and he worked on a gene of Anamnaceae, and he was sequencing TurnLF, a very widely used plastic marker. And what he found actually that this gene is here, fell outside, which is an Anamnaceae, it fell outside the family because the common ancestor of the family is sitting here. And we saw this and I'm like, what is going on here? This is so bizarre, Anamnaceae. I mean, 100% Anamnaceae species falling out of the family. I'm not going to talk about the details, but it turned out actually that we accidentally hit a parallelis copy, and when we deliberately searched for that parallelis copy, we got this tree, because you see many of the names here also appearing here. So this is TurnLF copy 1, this is TurnLF copy 2. As I said, a small cautionary tale. Usually you should be fine when you say, as I just said, my plastic genes are sitting on the same molecule. There should be no conflict. It almost always works, but not always, as we found out. And concatenation is still, of course, for very good reasons, let's be clear, for very good reasons, is still a preferred way of, an often published way of dealing with data. As in this paper that with the Thai student and Anna and we and several other colleagues published earlier this year, where we had 3 plastic markers and ITS, so from nuclear ribosomal DNA, and yes, we concatenated because we first established that there was no well-supported conflict amongst these 4 markers. Right, so, and many papers have been published even until very recently where this idea of data concatenation is still being used, such as, for example, in this paper with lots and lots and lots of data on ancestral phylogeny, 17 genes, 640 species, published by Doug Saltis' first author from Gainesville, Florida. And the usual suspects are there as well, including Mark Chase. But the point is, they still concatenated and you would think that if you put in 17 genes, so that's so much data, that you would finally arrive at a fully supported tree. But no, it's only 87% bootstrap support for that node. Which, given the fact that they put in sequences for 17 genes, that is quite a poor bootstrap percentage. But there's a good reason for that. And in a way, they also realize this in this paper when they say, however, the 17 gene is it's a significant improvement. Actually, they voiced a kind of optimistic, and probably might even think that if you just keep on putting in data, even at 87% bootstrap support might improve. But I think it won't. And the reason is this. This is a very nice paper. It's already 20 years old. It's on fungi. And this is for a group of yeast-like fungi for a clade. And the authors sequenced 106 fungal genes. And they did find that amongst these 106 genes, there was good incongruence, meaning, a gene could tell a different phylogenetic story compared to another gene. Yet, they say that larger concatenated, that's the word again, concatenated data sets have very powerful have been very powerful and persuasive. So they still advocate the concatenation approach. And this is I think by now you kind of feel in my tone, that's something wrong with concatenation. And this is actually, I think, the worst example, where they have 59, not plastic, but nuclear genes. They still concatenate them. Every biologist should know that you have such a high risk of running into incongruences when you look at individual nuclear genes, meaning individual nuclear genes can behave differently from some genes being upstream or downstream in the nuclear genome. So, and why is concatenation then a bad idea? As I said, because then you're adding conflicting phylogenetic signal and just throwing that all in the mix, in one single analysis. And here's the biological reason why that is not such a good idea. And it has to do, it's not the only reason, it's one of the, it's a very important reason. It has to do with how genes behave within organisms. Because, even in us human beings, it's not the case that all of our genes are inherited as one nice package onto the next generation. I'll say one more thing about that in a minute, but let me first focus on this one here. So here you see a simple phylogeny. So time goes from here, long ago, to here, less long ago. These lines indicate separation between species. So here you have a speciation event. So here's an ancestral species. Here a speciation event. So species 1 and 2 evolve. And here's the next speciation event. So this species then diverges into these two species. Now look at the colors. So the colors are individual genes. And this is a realistic scenario. Yeah.
These tiny dots are individual species, you are saying?
Genes.
Individual genes. These tiny spots there in the graph, these are individual genes.
Yes.
Okay.
Yeah. And so what you see here is that if you look at the genes, then purple here and purple here. So based on the gene, you would say these two species are each other's closest relatives, but they're not. Because these two share a more recent common ancestor, right? But so the organismal tree is conflicting with the gene tree inside. Now, and this phenomenon is known as, incomplete lineage sorting. There you have it. And the risk, or the probability, I should not risk, probability of seeing incomplete lineage sorting is mainly dependent on two things. Don't bother about the details of this formula, but there's two important factors in here. It's time, T, and population size, N. So the shorter the time between subsequent speciation events, the higher the probability that you're going to see this effect of incomplete lineage sorting. So
that means those species were in between the speciation. That's why we are getting these kind of results.
well, the thing is, because the time between subsequent speciation event is so short, the genes within the newly evolved species haven't had the time to sort of settle, to put it like that, to come to some sort of equilibrium, because immediately they're entering the next speciation event. So, and the other thing is the population size too. So the larger the populations, also the higher the risk of seeing this phenomenon. And I just actually talked about us, human beings, but there are good examples. So, I mean, we know that chimpanzees are the sister group, so are the most closely related species of us, Homo sapiens. But there are genes in, let's say, the genomes of hominids that will tell you that, I didn't indicate, but that the gorillas are actually our closest relatives. But that's the same phenomenon. Gorillas are not our closest relatives, but there are some genes which have which have been subjected to this incomplete lineage sorting. Now, it's this incongruence between individual genes that make concatenation of data highly, highly troublesome. And therefore, new methods have been developed, which are called coalescent methods. Coalescent phylogenetic methods have been developed, and this is a cartoon of one of the papers by Madison and Knowles, one of the early papers about coalescent methods, showing, it's a bit of a cartoon, but showing a species tree. So the species are A to to H, and then all of these lines are individual gene lineages, but showing that you actually can have gene conflict, but in the coalescent model, these conflicts can be reconciled into one single species tree. And this is if you ever want to read a really lovely paper about phylogenetics, about the concepts of phylogenetics, do read this paper by Scott Edwards, an American zoologist, who actually very nicely explained in this paper from 2016 that whereas we have, historically speaking, in phylogenetics, we have mainly been using concatenation methods. This is actually a special case of a multi-species coalescent model, which yet again is a specialized case of an isolation, migration, and network coalescent model, which is more like a population genetic model. And so this nicely shows that it's all right to use concatenation, but as I said earlier, only when certain conditions have been met, and in this case mainly the condition of non-incongruence amongst the phylogenetic signal of the different genes. Just an example, because we're dealing here with a phenomenon that is not special for plants. This happens in any organismal group. So any fast radiation, so not only flowering plants, but in this case also mammals, there you see exactly the same phenomenon, which is what this paper from 2012 was showing, and where Scott Edwards was involved in as well. So also, the early evolution of mammals, it's a similarly problematic, similarly challenging scientific problem, to resolve the phylogenetic analysis here compared to flowering plants or tetrapods. I mean, there's so many of these radiations. Hypermia, of course, I mean, in any level you see these variations. I'll just skip this a few of the time. So this has been applied to the early evolution of flowering plants as well. And I want to go on to an example from my own work. This is a former student of mine, Yuri Strijk. And with Yuri and a number of Ananaceae botanists, we published a complete genome of this fruit, originally from South America, Annona muricata, soursop. I guess it must be cultivated here in India as well.
Yeah,
Yeah.
in our garden it is available.
Yeah. So 2 years ago we published the high quality chromosome level genome of this species, and it's sort of derived from that this year a book chapter in a book called Underutilized Crop Genomes. But what I want to show, and it's actually from the book chapter, is I'm actually going to focus on this tree. So here here's our new genome, well, 2 years ago, right? So the Annona muricata genome, and we compared this with genomes of 12 different angiosperm species. So we had 13 complete genomes. We derived 689 nuclear single copy genes from it, and we concatenated all those genes. So we deliberately did something that we should not be doing, but we wanted to see what will the results be. And of course you get a single tree and it's nicely supported. But if you then start looking, like teasing apart the 689 individual genes, then we saw, and I'm going to focus on the left-hand picture side of the picture, then we saw that there are actually 3 different answers. So here you see 3 different phylogenies simplified with Umbrella, monocots, dicots, and magnoliads. And so 4 of the major groups of flowering plants. And you see there are genes that will tell you that actually the magnoliads are sister to the new dicots. Importantly, and that's what green and yellow indicate, not as opposed to red and blue. Green and yellow indicate a fairly, well, green very good and yellow fairly good statistical support for this relationship. So it's not a spurious result. And so there are genes well supporting this scenario. But there are other genes, amongst the 689, that say that magnoliads are sister to monocots. And then third, there's a number of genes that support this scenario where magnoliads are sister to monocots and dicots combined. Incompletely and insulting, that's what we're looking at again, but it's causing conflict amongst the individual genes, in which case you should not concatenate them, but use coalescent methods. Right, lots and lots of papers nowadays are published with many single copy nuclear genes. Anna talked about that a bit already, and actually she and many co-authors published earlier this year a paper using exactly that kind of methods, where we used the angiosperm 353 kit to produce gene sequences, and they were analyzed with coalescent methods. So this is a resulting a figure with 2 resulting trees from that paper. So, and now I'm going to wrap up slowly. So here's a question. Perhaps, I don't know, some of you might have been worrying by now, like, oh boy, do we now all have to go and use, these methods? Which, yes, they are expensive, often, Do we all have to do that? No, you don't. So I'm going to reassure you with an example that you can also still use good old-fashioned Sanger sequencing and still get meaningful results. And I'll come up with some sort of research strategy. It's not the strategy, it's just an idea that I thought of earlier today when I was thinking about this presentation. And so suppose you're working on a group of plants, a genus or a family, or in this case, Hypermia. And so this paper, well, in our case, in the case of Hypermia, this paper is there already, and this paper actually did two things. It did use HiveSeq data, so high throughput data, but it also had a second data set, which was a data set of over 1,200 accessions, not species, but because many species have been represented multiple times, but lots and lots of taxa, but only using ITS. Again, it's just a small part of the nucleosomal DNA. And this is a wonderful tree also for the project of Hannah, and we're co-supervising two PhD students from Kenya working on Hypermia. And actually this tree is really cool. And of course, it's only ITS, so you will see that, many of these nodes here don't have fantastic support or not even support at all. Well, that's the price you pay when you use, a rather small amount of data. Or here you will see a polytomy, so this part of the tree has not been resolved. But that doesn't necessarily need to be a problem, because in the case of the two students from Kenya, we found this very nice clade of 10, 11 species, all from East Africa, and they all share a particular biology. So, and this is a mature fruit and a young fruitlet of one of the species, Hypermia longijuba. So longijuba is sitting here in this clade, and these are some pictures from Vierberg last year in Kenya. So at least some of the species here have storage roots, like the sweet potatoes, and they are edible. So here are two students digging up storage roots of Hypermia longijuba, and here it's been dug up. So here we've got it was quite large actually. And here it's again, and then later that day we were very happy with the sweet potatoes. We took lots of pictures. Later that day, we collected it again, but then in flower, and it turned out to be a night flowering, whole cloth pollinated, sweet scented flower. Absolutely fantastic. And it turns out, if you look at the literature, that all of these species actually have this kind of flowering, behavior, night flowering, sweet scented, hawk moth pollinated. So the point I want to make here, and that's going to be, then I'm really going to be silent. Even using one marker, ITS, we were able to, not we, the Oxford group, were able to demonstrate there's a clade actually, which is well supported, with 10 or 11 species that all share a similar biology. So that could be, a take on a project. Well, of course, this is the first condition. Your taxonomy needs to be up to date. You need to know what species you're talking about. But then you could use a summer sequencing approach to come up with a larger framework, as we're having here. And then you could see which clades are really interesting, like in this case, which clades have an interesting biology. And then, if you want, you could, to further dive into that clade, you could apply high throughput sequencing for a more detailed analysis of a smaller clades. So combining big picture, focused approach, and of course, avoiding that you really spend too many rupees on your project, because, well, of course, money is always restricted, one of the restrictions in our field, for any one of us, I guess. Okay, I want to leave it with that. Thank you.